Welcome everyone to the closing keynote of our virtual Friends for Life Indianapolis conference. Ed Damiano will be presenting an update on the iLet Bionic Pancreas. I'd like to thank all of you for sticking with us and tuning in during these very interesting times in which we are living. And I would also like to thank our sponsors, Lilly Diabetes, Dexcom, Insulet, Tandem Diabetes Care, and the American Diabetes Association for supporting us in our first virtual conference. As part of this, you have the opportunity to enter questions on the right-hand side in the control. We will save questions for the end, but please, as you think of them, type them in and we'll make sure we get to them at the conclusion of Ed's presentation. And without further ado, I'd like to introduce Ed Damiano to present our closing keynote. Ed, it's all yours.
Thanks, Jeff. All right, can everyone see my screen?
You're good to go.
Okay, thanks, Jeff. Trying to get this out of the way so it's not. I assume this is not obscuring the screen for anyone else.
Well, we see your mouse. If you just push it off to the side, you'll be good.
Okay, very good. All right, that's good. Super. All right, thanks, Jeff. All right, folks. Well, thank you for hanging in there and participating in this conference virtually. I hope you all are well and safe and out of harm's way and hunkering down in your residence and enjoying the Friends for Life conference this weekend. I'm hoping I can give you some good news, some positive news at the end here, and continue to sort of move this technology forward and get it to people with type 1 diabetes as soon as we can. It was about 6 years ago when I jotted down this Go Bionic mission of ours, and this particular emblem you see here on the screen. And despite all the trials and tribulations of COVID that we're all facing around the world, we are still committed to Go Bionic, and I hope that we can give you some reassurance that we are moving as hard and fast as we can despite the setbacks that we're all facing. So I want to get started, but before I do, I just want to just share my disclosures with you all. So I am, oops, that's another one. So I'm a co-founder and shareholder of Beta Bionics, and I'm also a board member and a member of the management team. So I wanted to mention those conflicts of interest as well as this other one that's oh so important to me, and that's my son David. So many of you may be familiar with the work we've been doing developing a bionic pancreas for the past 15 or 20 years or so. This was work that started in my lab at Boston University. A little bit before that, in fact, at the University of Illinois, when my son David was diagnosed with type 1 diabetes at the age of 11 months. David's now a junior at Boston University doing online classes like the rest of the world is. So he's home safe and sound and finishing up his junior year through Zoom windows and the like. But this is a picture of David when he was just a few months, just a few years old, just a couple years after he was diagnosed with type 1. And, we have been committed ever since then to try and build a technology that can manage his diabetes automatically. So we were really committed from the beginning to building a system that had a real turnkey simplicity, that anyone could use, that everyone could use, that required nothing more than your body weight to get started, no expertise of an endocrinologist or some other subspecialist, a diabetologist. This is something we envisioned could be started by a physician at home, in clinic, a primary care physician, endocrinologist, a subspecialist, remotely, any number of ways you can imagine. Because of the simplicity of the device, many more prescribers could use it and many more patients could benefit from it. So that was our commitment from the beginning, and we've really held fast to that commitment. In the meantime, while we've been building the technology, first testing it in animal studies and then human trials, and then after that, inpatient first and then outpatient trials. We've been working away at this system and making it better and refining it and improving it. David, all the while, has been growing and taking care of business himself. And so as he's done that, he has progressed to a high school junior. His sister there, the smaller of the two there, is graduating last spring, also from Boston University. So we realized that if you feed David and give him insulin, he will grow. And here's the evidence of that. He's done a wonderful job of navigating diabetes and college over the past 2.5 years. So what I want to do today really is provide you all with an update on where we are with the iLet Bionic Pancreas system, give you a sense of what that technology is, where it came from, how it works, and how well it works in clinical trials that we've conducted over the years, and also, of course, give you a sense of where we are in the context of COVID and the impact that has had on our clinical trial plans. So before we get started, I just would like to make a couple comments about diabetes management. Of course, you're all, experienced living with type 1 diabetes, having family members with type 1 diabetes. But nevertheless, despite how closely we are all connected to this condition we have, it's really helpful to understand this problem in the context of the population at large in the United States. And the data you see here come from an epidemiological study done in 2015, looking retrospectively at about 3 years worth of data from 81 expert clinics across the US. So these are data taken mainly from endocrinology clinics and diabetologists across the US. So this really is a look at what the best endocrinologists are able to achieve in managing people with type 1 diabetes in the US. And what you're seeing is the number of patients who are meeting the American Diabetes Association goal for therapy, shown in green on this bar chart, and those who are not are shown represented in yellow. So you can see we're really falling short of the mark here across adults and children alike. In fact, it's even worse in children now, given that in the sense that the goal for therapy has been lowered now by the ADA in children down to 7%, just like it is in adults. So even fewer children are meeting the revised goal for therapy than when I prepared this slide just a few months ago. So what you see, of course, is that the best controlled age groups are the mature and older adults, but still, even there, fewer than 1 in 3 Americans in those older age groups are meeting the ADA goal for therapy. And it's particularly bad in children. Teenagers and young adults are fewer than 1 in 5. Meeting goal for therapy. So despite all the technology, despite in terms of the insulins that we have, the insulin analogs we have, the ultra rapid insulins, continuous glucose monitors, and insulin pumps, we still are not coming anywhere near meeting goal for therapy. But as I mentioned, these data are taken from expert clinics. So as long as I've been showing these data, I've always attached the caveat to this that these data are really looking at the U.S. People with type 1 diabetes in the U.S. through rose-colored glasses. In fact, it's probably quite a bit worse than this we've been thinking. And we now have reason to believe that that statement is true with some compelling epidemiological data that was published just last year. So what you see here is the same data I just showed a minute ago, but it's just localized to the adults 18 and older. So we're just focusing on the adult cohort of that study. That was published in 2015, where you have about 7,600 subjects, all seen by endocrinologists and diabetologists between 2010 and 2012. The same group of investigators visited this data again and found that things got no better, in fact, worse in some age groups when this data was looked at in many of the same subjects between 2015 and 2018. So despite the advent and the increased usage of continuous glucose monitors, we haven't seen significant improvement in glycemic control in the same similar cohort and across the same 81 expert clinics. But more recently, data taken from 2015 study between 2015 and 2016 showed across primary care and endocrinologists, patients who hadn't seen an endocrinologist for over a year, who are likely getting their care from primary care physicians in a much larger cohort of about 30,000 patients, show that when you cast a broader net and look at people who aren't just being visited by these 81 expert clinics, who aren't simply seeing the best diabetologists and endocrinologists in the world, you'll see all three of these categories drop. So when I advance to the slide to this larger data set of over 30,000 adults, you see in every one of these categories, the number of people meeting goal for therapy is actually lower than the T1D exchange data from those expert clinics, as you might expect. So in this cohort, about 40% of them did not see an endocrinologist in the whole year. Over the whole year period, the data was collected. So we're seeing a situation here where in the young adults, approximately only 1 in 8 are achieving goal for therapy, and fewer than 1 in 5 mature adults and 1 in 4 older adults are meeting goal for therapy. So we have a long ways to go here. And the real problem is not that the patients are not compliant or the physicians are not good enough. The problem is that managing diabetes with the tools we have today just doesn't suffice. The tools are not refined enough instruments. They're too blunt. They're simply not able to help people manage diabetes well enough to get the kind of control that we really need to achieve for everyone with type 1. And that's really where the Bionic Pancreas comes from. You can see that data set I just showed you in a slightly different format. The data you see in this slide shows both of those data sets now superimposed, where the solid line traces the T1D exchange data from the expert clinics of about 7,600 adults, and the dashed line traces the control across different ages in the larger 31,000 cohort, many of whom didn't see endos. So you can just see at every age across the spectrum from 18 to 85. We're doing much worse out there than expert clinics can achieve. Even though those aren't doing so well, we're seeing even more challenges when you take in account of primary care. So the reason this is so important is that a huge number of people, we don't know precisely how many, but a huge number of people in the US with type 1 diabetes simply have no access to endocrinologists. Not to say that having access to endocrinologists dramatically moves the needle, as you just saw. Instead of a fifth of people meeting goal for therapy, maybe it's more like 30% instead of 20%. But still, even then, the vast majority of people are too far away from these major endocrinology clinics, as this map shows. So the Ozers published a paper just recently showing the county by county coverage by primary care physicians. So if you take a look at every county over 3,000 US counties shown in the lower 48 here, you see that each county drawn in blue has at least one primary care physician, general practitioner, internist, family practitioner, pediatrician, and the counties that are drawn in white have not even one. But the very few and sparsely distributed counties that don't even have one physician, nevertheless, primary care physician, nevertheless have a county adjacent to it, north, south, east, or west, at least one county adjacent that does have a primary care physician. The point being, you don't have to drive great distances in this country to find a primary care physician. On the other hand, if you take a look at this demographic distribution across pediatric and adult endocrinologists, look at how sparse the distribution becomes. You can see vast tracts of land across the US have huge numbers of counties with no pediatric or adult endocrinologists anywhere to be seen. There are huge distances people would have to drive across many of these counties in the US to find someone to find an endocrinologist. So ultimately, you see the impracticality of expecting that endocrinologists would be able to support all the people with type 1 diabetes in the country. Consequently, we need to build technologies that everyone can use, whether you're a subspecialist or a primary care physician. So even technologies that automate insulin therapy to some degree, so-called hybrid closed-loop systems, even those systems are likely only going to be deployed in these counties drawn in blue here on the right side of your screen, where there are endocrinologists. So even as those technologies do improve the standard of care, they're simply not going to be able to get out to most of these counties drawn here in white, because they still will be too complicated to get started and to maintain by primary care physicians who aren't expertly trained in the use of these technologies. This is where the Bionic Pancreas comes in, as I mentioned. This is a technology that was designed from the beginning with turnkey simplicity that does not require physicians know about basal rates or carb to insulin ratios, insulin correction factors, insulin sensitivities. It doesn't require patients to carb count. So physicians who are unfamiliar with all of those various aspects of pump therapy, which are currently required of all the hybrid closed loop control systems that are out there or that are being that are under study now, those systems are likely still going to be unavailable, and we're still going to see a tremendous throttling, a tremendous limit to the deployment of those technologies broadly across the US. The Bionic Pancreas, on the other hand, is a device that has that simplicity I described. There's no requirement to count carbs. There's no carb to insulin ratios that you need to program into the system. There's no basal rates. There's no correction factors. You type in your body weight, and the system then begins to adapt and autonomously learn, lifelong learning in real time, every 5 minutes of every day, continuously. So the idea is we have a system that as a machine is capable of autonomous lifelong learning, requiring nothing more to get started than your body weight, and then continuing to adapt ever onward. And then it adjusts as you change, as your insulin requirements change, as you have increased insulin sensitivity due to physical activity or certain kinds of intercurrent illness, or if you have increased insulin resistance due to inactivity or due to other kinds of intercurrent illness, the device adapts and follows your ever-changing insulin requirements. And that's how the system was initially contemplated. It took us years of development to figure out how to build those mathematical algorithms that determine just how much insulin and glucagon to dose to achieve that kind of control. But ultimately, we have built a system that is robustly adaptive, as we've shown across children and adults, using the system in clinical trials, that gives us lots of confidence that a system like this truly is possible, and that all these counties drawn in white here could ultimately one day benefit from a system with that kind of simplicity. So that's where this cute little cartoon comes from. This was conceived a long time ago. A woman named Haiti, who has type 1 diabetes, drew this up for us. It's a lovely cartoon, just sort of making a comedic depiction of the Bionic Pancreas, where you have a continuous glucose monitor that sends data every 5 minutes into our device, which has embedded on it mathematical dosing algorithms, so mathematical equations, if you will, that make therapeutic decisions every 5 minutes to determine how much insulin or glucagon to deliver. Those mathematical equations then dictate how much insulin and glucagon to deliver through a pump. As you can see, she's drawn those as little well pumps. And ultimately, the idea here is that this system would replace, or at least as much as possible, emulate the function of the endocrine pancreas for people with type 1 diabetes, and replace all the management requirements that they're burdened with to take care of diabetes, and take that over itself. So it's quite apropos that we call it a bionic pancreas when this cartoon, of course, references the $6 million bionic man, as many of you who are older in the audience may recall. So we started building a system that took those early forms of the mathematical dosing algorithms and put it on a laptop computer. So my PhD student, when we started working on this problem around 2002, about 18 years ago, was named Feeis and he still works on the project with us today. So he leads our clinical and regulatory affairs at Beta Bionics, which I'll talk about in a little while. He wrote the initial code that we used to test the system in pigs. We built a system that ran on a laptop computer. It interfaced with a continuous glucose monitor. The first continuous glucose monitor we used back then was the Abbott Navigator, which was approved in 2008. And we tested these CGMs in head-to-head studies across a variety of subjects in human trials before choosing the Navigator back then as the go-forward CGM. So we built this system that worked with the Navigator. Initially, we used Deltec Cosmo pumps, but then we moved and migrated over to Insulet Omnipod pumps before ultimately switching to tandem pumps and then finally building our own platform. So this system was quite clumsy. As you can see, it hung on an IV pole. There was a venous blood glucose monitor that was strapped onto that system so we could measure in the inpatient setting blood glucose every 5 minutes. We first did pig studies with this device between 2005 and 2009 at Boston University. And then between 2008 and 2012, we brought the system into the Clinical Research Center at the Massachusetts General Hospital in collaboration with Stephen Russell and David Nathan. And we did several experiments, initially in adults, but then ultimately in adults and adolescents. Our last inpatient study at MGH was done in 2012, the end of 2012. And that's where we were really able to start testing our learning software, our autonomous learning system, so that we really were initially building a software that worked quite well in adults, but their insulin requirement was not very, very different from one patient to the next. But when we brought adolescents into the clinical research center, of course, they typically use a whole lot more insulin than adults do. We really found our system was challenged to give enough insulin in those patients. So we really had to go back to the drawing board around 2010 and start building a system that was much more robust in terms of its ability to adapt and learn about changing insulin needs or higher insulin needs in one person versus another, despite the fact that they both might have the same body weight initially. And when you initialize the system, it has to be able to depart from that and pretty robustly. So that was the work of 2010 to 2012 with the laptop system. We were also testing continuous glucose monitors throughout that process all the while, and found that the Abbott Navigator was just superior to every other CGM we were using until 2012. Second half of 2012, we began testing the 4th generation, or the so-called G4 Dexcom CGM, and that system was as accurate, we found, as the Navigator. The Navigator was pulled from the US market around 2011, so we didn't have a path forward with that CGM anyway. So we were really fortunate that the folks at Dexcom had continued to innovate and continued to advance the quality and accuracy of their sensor. So by 2014, by 2011, we were able to start building our own system. It ran on an iPhone, and we initially integrated it with the Navigator, but as that became harder to come by, and we started to see the data coming out of our own experiments that the G4 Dexcom was really as accurate as the Navigator. By 2012, we built this system you see here in the middle of the screen. So we'd gotten the system off of the mathematical dosing algorithms that were running on the laptop were now embedded in an app of our own design, and ran on an iPhone 4S, which was the first smartphone at the time back in 2011 that had a low energy Bluetooth radio as opposed to that classic Bluetooth. So it used a lot less power than the previous devices that used standard Bluetooth. And the Tandem had just received FDA approval of their first insulin pump in late 2011. So we started working with the Tandem engineers to integrate our app running on the iPhone 4S with their Tandem pumps. And ultimately what we had was this rather a clumsy system, but nevertheless, it was something you could put in your pocket. So for the first time, we could get out of the clinical research center, out of the inpatient setting, and into a real home use and outpatient setting. So between 2013 and 2018, we did a number of experiments with this system. We could be much more productive in terms of the number of experiments we did in a year. And over a 6-year period, we conducted a variety of experiments in adults and children with type 1 diabetes, both in the outpatient and summer camp settings and in the home use setting in adults, at multiple clinical sites where MGH led the way in most of those studies. But Stanford was also involved in testing the system back then, as well as the University of North Carolina at Chapel Hill and the University of Massachusetts. We continued to test the system in other populations, in adults and children with congenital hyperinsulinism, people who make too much insulin. They don't have diabetes, they actually have kind of the opposite problem. They secrete far too much insulin, and they suffer from chronic low blood sugars. This was an inpatient study that we were able to do in children and adults with the system. We got FDA approval for investigational use of this device in cystic fibrosis-related diabetes in adults with CFRD. And tested it in collaboration with Stephen Russell at MGH and Melissa Putman at Boston Children's Hospital. And we also did some preliminary studies in adults with type 2 diabetes in the home use setting, as well as in adults with who are adult post-bariatric surgery patients who often suffer from hypoglycemia, who sometimes suffer from hypoglycemia. So it's a rare condition, but nevertheless, one that is fraught with lots of hypoglycemia. So some of these populations, the CHI, the CFRD, and the post-bariatric surgery patients are orphan conditions. They're small populations, but they have a great need for a device like this. So this system really helped us improve the device, improve the algorithms, the mathematical dosing algorithms. We were able to test the device in all these different populations. We were able to make refinements to the algorithms and improve how well they adapt and learn and make them safer and more effective. We were able to study the device operating at different glucose targets. We were able to study how the device worked with only insulin or with only glucagon. And so we learned a lot about the system, and we have a lot of confidence in what it's able to do in terms of lowering mean glucose and simultaneously reducing hypoglycemia in the bi-hormonal configuration, and how well it works also in the insulin-only configuration. So we can be pretty predictive, we found, with how well the system will control blood sugar in a population of people with type 1 if you use sort of a middle set point, our default set point in insulin-only configuration versus a lower but default set point in our bi-hormonal configuration. So we can be really essentially almost predictive now of how well the system performs in these different configurations. And that really is due to all the clinical research that we did over these years. But around 2014, as we started to survey the landscape of insulin pump manufacturers out there, it became clearer and clearer to me and to some of the other folks we were working with that at the time that we weren't really going to be able to do this without building our own dual hormone infusion system. So as I mentioned, our system can deliver both insulin and glucagon, or we can configure it just to deliver insulin or just to deliver glucagon. So we really need a dual chamber system, a pumping system that has an insulin chamber and a glucagon chamber that can be independently actuated with two independent motor drivetrain units built into the same unit. So as nice as it was to be able to do these home use studies, we really needed to build a much more compact and practical portable system. Beginning in 2014 in my lab at BU, we started building what we call the iLet Bionic Pancreas system. We built a first-gen system, which was really just a prototype system we could test in the lab. And then we built a Gen 2 iLet system, which we revealed for the first time at the 2015 Friends for Life conference in July in Orlando. And that system we actually tested in pigs. So we did some animal studies with that system. It was quite a bit larger than the system we use today, but it was still smaller than the iPhone system you see here. And then in 2017 and 2018, really starting in 2016, all the way through 2018, we built a third generation iLet, which was smaller than the Gen 1 and Gen 2 iLet systems. And they were all dual chamber, of course, but it was smaller, more compact, and much more practical device. And that was the first iLet system we brought out to people with type 1 diabetes in a home use setting. And then ultimately, while what we call the Gen 3 iLet studies were going on in 2018 and 2019, we began building at Beta Bionics the commercial version of the system, what we've been calling internally the Gen 4 iLet. And that system is really truly a very elegant device. It's extremely small. It's smaller than the first iPhone back in 2007 in terms of the volume of this device. It's really small and easy to tuck away. Despite having two chambers and two motors and two drivetrains, it's still an extremely compact unit. It's only 15 millimeters thin. It's got a very nice capacitive touchscreen with a good backlight, so you can see it under all different kinds of lighting conditions, under low lit conditions or very, very bright sunny days. It's a really easy display to read. It's a convenient system in that it doesn't have any replaceable batteries. It has rechargeable batteries. And specifically, it's an inductively rechargeable system, so you don't need to plug it into the mains to recharge it. You can just lay it on an inductive charging pad, the same charging pad that is used for iPhones and Android devices is the same device we use to recharge the iLet. The battery lasts quite a long time. It's we were able to get the battery life of about 5 days on the system under normal usage. And it takes about an hour to charge it up, 1 to 2 hours to charge it up. So if you charge it every 15 minutes every day, you'll never have battery problems. So we've really made it as convenient as we can, given the fact that it is still a device that's hooked up to you with 1 or 2 tubing sets, infusion sets, and a CGM. You still have to change the cartridge or cartridges when they're empty. And you have to charge it every, so often, as I mentioned, once a day for a few minutes. And you have to change the infusion sets and the sensor on regular intervals. So I'm not suggesting it's not a burdensome system in that sense. There's a lot of maintenance that goes along with a device like this, no doubt. But it does take the diabetes management off your plate, right? So that's really what we're trying to do, is give people really good clinical outcomes, much better than the standard of care can achieve, for far more people, prescribed by many more providers than can use current technologies. And to do so with a simplicity that makes access of that sort really possible. And that's really what's essential to scale a system like this. To truly bring a system like this to scale, you need to have that kind of simplicity. So we need to have really good clinical outcomes has been our goal. Our clinical data support that we can do that in both the insulin only and even more so with the bi-hormonal configuration, but also to do it with the kind of turnkey simplicity we've been committed to since the very first pig experiment we ever conducted back in 2005. So you can see here with the Gen 3 iLet system, at least, we haven't begun testing the Gen 4 system. That's going to be the work of this year. But the Gen 3 iLet system was used in 2018 and 2019. And it was tested in the insulin-only configuration in adults and children with type 1 diabetes in the second half of 2018. We used it again in a study sponsored by Novo Nordisk to test it with ultra rapid insulin, comparing it to rapid insulin. So we used Feeis and compared it with just Novolog against Novolog to see if we could get better control with the system under those circumstances. And finally, we did a bi-hormonal study testing the system for the first time with a 4 milligram per ml concentration of dasiglucagon, which is a stable glucagon analog, which tries to mimic the kinetics of the pharmacokinetics of human glucagon, which all our studies prior to 2018 or so used regular human glucagon, which is not chemically stable and doesn't really have a commercial path forward as a chronic use drug in this application. But it did work well when you reconstituted it every day at the point of care. If you change that glucagon daily, we got really good control and we could really prevent hypoglycemia much better than insulin-only systems are able to do. And still get even lower glucose, mean glucose because of that. But there's no commercial path forward. There was no scalability of that because it's not practical to use a chemically unstable glucagon. So instead, we really were hoping for the pharmaceutical industry to develop stable alternatives to human glucagon, either stabilizing human glucagon like Xeris Pharma has done with a different excipient. Instead of using water, they use dimethyl sulfoxide, or making a whole new molecule like Humalog and Novolog is not a human insulin molecule, it's an analog of human insulin. Zealand Pharma has built a stable pumpable molecule that they call dasiglucagon, which is very similar to human glucagon. It's got slight differences in the peptide that give rise to really high stability in water. So that drug is even more stable than the dasiglucagon is even more stable than Humalog or Novolog is in solution. So it's a totally practical solution in a pumping environment. And all of our in-use compatibility studies bear that out. And it has very similar kinetics, absorption kinetics, to what we saw with the human glucagon. It's very rapidly absorbed and very rapidly cleared. So it's a really wonderful drug candidate, and it is we are phase 3 ready, or Zealand is phase 3 ready, together with the iLet system to begin our final pivotal trial with that drug in our system. So that is something that we're hoping to begin either at the end of this year or beginning of next year. So that's a very encouraging progress that we've made there, and I'll talk a little bit more about that in a moment. And this just shows you with the going back to the iPhone system for a moment. These are clinical results that we have obtained with the iPhone system back in 2013 through 2015 in our first 3 outpatient and home use studies with that platform. And what you see here is you can see these are different 3 different studies. One was the teenagers in summer camp, 5 days on the bi-hormonal Bionic Pancreas and 5 days on the usual care in random order. The middle panel shows preteens also in that same summer camp at Camp Joslin and Claire Barton camps in central Massachusetts. They were, again, 5 days on the bi-hormonal Bionic Pancreas and 5 days on their own usual care. And then we have data from our first true home use study, where we had 39 adults living with the living with the Bionic Pancreas, bi-hormonal Bionic Pancreas, for 11 days on their own usual care and 11 days on the bi-hormonal Bionic Pancreas in random order. And in that study, they were able to just go about their business and live their normal lives and go to work and drive or take the public transportation or whatever to get go about their activities. They could be as physically active as often and as vigorous as they wanted to. There was no restriction on diet or activity. So we were really getting a good look at how this system might perform in the wild out there. And as you can see, on each of these plots, we have a bunch of blue lines. Each blue line corresponds to one subject. And where each of those lines ends, terminates on the left side of each of the 3 plots. It corresponds to the mean blood sugar the mean glucose, or the CGM glucose, that each of those subjects obtained on their own usual care in each of these 3 studies. And then where those blue lines terminate on the right tells you how each of those subjects, what their mean CGM glucose was across the study period on the bi-hormonal bionic pancreas. Again, this was the iPhone system with the 2 tandem pumps, and the G4 Dexcom was the input to the system. But it was our algorithms from the beginning that were being used in all of these studies. And another thing to note about these plots are the red circles. The size, the diameter of the circle corresponds to the time the average amount of time each subject spent below 60 mg per deciliter. We've since moved to 54 mg per deciliter going forward, but for the studies, most of the studies we've done, we were using 60 mg per deciliter as that metric. And you can see on the left-hand side of some of these plots are some pretty large red circles, which would correspond to a great deal of time below 60 mg per deciliter, and pretty concerning level of hypoglycemia in some of these subjects. But generally, you see that when you look at the right-hand side of each of these plots, the circles are smaller, and the mean glucose levels are generally lower for most subjects than they are on their usual care. And in many instances where their mean glucose is higher than on their usual care, their hypoglycemia is significantly reduced. So the device would favor taking someone who has really low blood sugar with a lot of hypoglycemia, and it would certainly prefer a slightly higher mean glucose in some of those cases with much reduced hypoglycemia. So it prioritizes, if you will, preventing hypoglycemia at all costs before lowering mean glucose. It still, nevertheless, does a really good job of lowering mean glucose. And as you can see here, in all 3 of these studies, the mean glucose was almost the same across each of the 3 cohorts, a mean glucose of about 140 mg per deciliter. Whereas you see that it's quite a bit higher under usual care. Remember, in all 3 of these studies, this is the same insulin dosing algorithm that we were using across the teenagers, adults, and preteens. And the only information the system got was at the beginning of the experiment was their body weight, and that was it. And there was no other input to the system besides the CGM every 5 minutes. So we get much lower mean glucoses for the cohort as a whole, significantly lower, significantly less hypoglycemia. Mean glucose is, again, all around 140, which would correspond, if you ran the study longer, to an A1C of about 6.5%. But also we found that the hypoglycemia was statistically significantly reduced, sometimes by about a factor of 2 or more, depending on whether it was the teenagers or the adults. The mean glucose seemed pretty insensitive to whether or not it was a teenage or adults or preteens. But the other thing I'll point to you, will call to your attention, is the standard deviation around the population mean. So that's the number after the plus and minus on each of these statistics. So you'll notice for the usual care on the left, the standard deviation around the mean, the amount of inter-subject variability, how much variability there is, looking how spread apart, how scattered the data are on the left side of the plots versus how tightly clustered they are on the right. The standard deviations around that mean are roughly around plus or minus 30 mg per deciliter. You see 27, 30, and 29 mg per deciliter for usual care in the 3 plots on the screen. If you take a look at the right-hand side, when they're on the bionic pancreas, those standard deviations are much closer to between 10 and 12 mg per deciliter. So they're about around 1/3 as much variability, if you will, relative to usual care. So we're able to take people who have a lot of variability across a cohort and dramatically reduce that, knowing nothing more than their body weight to start the system, and then letting that system adapt and learn autonomously how to better improve their glucose control and adjust to their ever-changing needs, while also lowering their mean glucose and reducing hypoglycemia. So these are 3 things that seldom go together. Reducing mean glucose in a cohort at the same time as lowering hypoglycemia. Those things usually are at odds, they're in tension with one another. And then also reducing intersubject variability is also particularly challenging to do that while doing these other things. So the fact that the bionic pancreas is able to do all 3 with a system that has this kind of turnkey simplicity is really promising for people with type 1. And here's just another way to put these data in the context of a much larger population. The bionic pancreas basically can be compared, if you will, to the standard of care. So if you have a look at the data that I've shown here, this is just the same data I showed at the beginning of the presentation. So what you see is the data from the T1D Exchange Registry plotted across age groups. So if you look at this red trace, you can see where the red trace lands in the middle of each of these gray shaded areas is the mean glucose for that age group. So you can see that the young adult population has a mean glucose around 200 plus or minus 55 versus 174 for the young adults plus or minus 37, 171 for the older adults. Immature adults were 174 plus or minus 37, and the older adults were 171 plus or minus 32. And you can see the red dashed line, which corresponds to the American Diabetes Association goal for therapy. And you can see just how poorly, of course, we're doing out there, just like I showed at the beginning. But this plot also adds that standard deviation, that error bar, if you will, on around each of the means shown by the red line. So there's a lot of variability in this cohort that I didn't show in that first slide. So that's really the problem here, is that not only are very few people meeting goal for therapy, but there's a tremendous disparity in the kind of glucose control people are able to achieve even within these age groups. So in each of our home use and outpatient trials, we always had a usual care arm. And so here I'm going to show you the usual care arm sort of lumped together in sort of a meta-analysis from all these home use and outpatient studies that I talked about with the iPhone system. And so when people were on the usual care arm of our studies, so as opposed to having thousands of subjects, now we're down to hundreds of subjects because these are coming from our own phase 2 trials. You can see that people did pretty well when they were on usual care in our own clinical trials, certainly better than the population at large. The mean glucose dropped down to 161, and that's a cohort of people 18 and older. So it's capturing all the age groups to the left here, but just in a small cohort and this is a self-selected group of people who are likely more engaged in their diabetes management. They're participating in a clinical trial, and they're probably participating and trying to do as well as they can while on usual care. So there's a few study effects here going on as well that make this cohort perform better than the population at large. But you can see that ultimately there's still fewer than 50% are achieving the goal for therapy. And they still have a large standard deviation around the mean, almost plus or minus 30 mg per deciliter. So there's still a lot of variabilities you saw from the previous slide in our usual care arm, even though their mean glucose is better than the population at large. And here you can also see the amount of hypoglycemia that we captured, about 2% of the time below 60 mg per deciliter. So that's still quite a bit of hypoglycemia. That's like 30 minutes a day being below 60. And so if you look, by the way, the folks on the left there that are shown on the left slide in gray, they had hypoglycemia too, for sure. It's just that data wasn't captured because they weren't wearing CGM, this epidemiological data set. So we don't know how much hypo they had, but there most certainly would be circles, just like we see under our usual care arm. We just don't know how big they are. But if you take a look at about 40% of people achieving goal for therapy under usual care in our trials, when those subjects use the insulin-only Bionic Pancreas, we see that nearly 50% would achieve goal for therapy. We see the hypoglycemia drop from about 2% to about 1.2%, so it cuts almost in half. And we see the standard deviation around the population mean also cuts in half. So instead of plus or minus 30, we're looking at plus or minus 14 mg per deciliter. So there's much more clustering of patients close to that ADA goal for therapy, which means they're doing nevertheless better, even if they're not hitting goal. And about 50% are achieving goal. And the mean glucose here is about 154 mg per deciliter in our insulin-only system, which would be an A1C of about 7%. So you're looking at an A1C that is right, the average would be right at the ADA goal for therapy, and about half of the population would come in under ADA goal, and with quite a bit less hypoglycemia than what we were seeing under usual care. And by the way, let's not forget that this is much, much easier to get this result in these people because they're not counting carbohydrates. They didn't have to know what their basal rates or carb to insulin ratios were, or sliding scale correction factors, as opposed to the people when they were on their usual care, they were working very hard to get that control. And nevertheless, they were getting worse control by every metric with much more intersubject variability and a tremendous more effort expended by the patients. When you add the second chamber and you bring the automated glucagon, microdose glucagon into the picture, here's results from the bi-hormonal system in this population. You can see that you go from about 50% of people hitting a goal for therapy to 90% of people hitting goal for therapy. So the mean glucose dropped from about 154 to an average of about 140 mg per deciliter. The standard deviation around the population mean stays small, right? It's still around 14 mg per deciliter, but the hypoglycemia drops below about 1% now in terms of time below 60. And now you're seeing about over 80% of people, you know, excuse me, I misspoke. So about, you're only above 180 mg per deciliter about 20% of the time. So about 80% of the time you're below, 180 mg per deciliter. So our time in range we see for the insulin-only system is typically about 70%, whereas with the bi-hormonal system we see that number is closer to about 80%. But I think it's really important to appreciate the standard deviation around the population mean. That number being so small really shows you how tightly clustered all these people are around the mean, which means that we're bringing many more people under the goal for therapy, which is what we want to do. So about 50% of people we think would hit goal for therapy with the insulin-only system based on these data versus about 90% with the bi-hormonal system. And when we bring these data to the pediatric population, we see the same kind of trend. So here's that, again, that epidemiological data set of several thousand children, teenagers and preteens taken from the T1D exchange registry. Large standard deviations around the population mean you can see the teenage population is performing just like the young adult population, very poorly. Mean glucose of over 200 mg per deciliter with about a standard deviation of around 50 mg per deciliter. So a tremendous amount of inter-subject variability. The ADA goal for therapy, just before we made this slide, was higher. It used to be at 7.5% A1C, which would be 169 mg per deciliter. The ADA has now lowered that target down to the same goal that they hold to adults. So I should update this slide to reflect that. But here's the usual care cohort in our outpatient diabetes camp studies with the Bionic Pancreas. This is the bi-hormonal Bionic Pancreas study. But this is the week when they were on their usual care and not on the device. And here you would see about 3/4 of them would achieve the old ADA goal for therapy. That number will be quite a bit less. If you look at the current goal for therapy, that number would be probably more like 40%. Like we saw with the adults. So the mean glucose is 162, again, a very large standard deviation of about 30 mg per deciliter, about 2.5% of the time below 60 mg per deciliter. And there was a tremendous amount of effort expended at keeping these kids in control to this extent during this trial, both of these trials. These are her summer camp studies in 2013 and 2014. When they were on their usual care, there was an endocrinologist on call 24/7. There were CDEs present all the time on camp. They all got their boluses for lunch, breakfast, lunch, and dinner. They never missed a meal bolus. They also got boluses for their 2 snacks every day. They had their finger stick measurements done about 7 times a day and once in the middle of the night if the midnight check warranted a 3 a.m. check. So these kids were under excruciatingly good control. And I emphasize the word excruciating. It was a huge effort. It's a totally non-scalable solution. And yet, when you see how well they perform when you put them on the bi-hormonal system, it's really humbling. Because despite all the effort that those healthcare providers could provide this small cohort at summer camp, they couldn't come close to the kind of performance that the bi-hormonal system was able to achieve in that same group under the exact same setting, same circumstances. About 100% of kids met goal for therapy. If we apply the new goal, it would be about 90% would have hit goal, just like in the adults. Mean glucose was again about 140, like in the adults, plus or minus 12 mg per deciliter. So again, a very small standard deviation. And we cut hypoglycemia from about 2.4 to 1.3% of the time below 60. So again, this would be about 80% of the time in range for the bi-hormonal system. So we're seeing the same kind of behavior in the kids as we're seeing in the adults with this system. And just the comparison of this with this kind of turnkey simplicity, the same device on the pediatric, pre-adolescents, on the adolescents, on the adults. Device doesn't really care across the age group. It's really quite remarkable that we can get this kind of control with this kind of simplicity. This is truly a scalable solution. This is something that can be enjoyed by this kind of technology by physicians across the country. No matter what their training is in diabetes management, they should be able to start a system like this and help their patients use it. So we published these results of these studies across papers throughout the past 10 or 12 years or so in the New England Journal of Medicine, in the Lancet, and in the Lancet Diabetes and Endocrinology. So it's been vetted through a peer review, extensive peer review process. We have a number of other studies that are under review or are going to be submitted soon for publication, where we've tested the system, as I mentioned earlier, across different glucose set points and targets, using the glucagon-only system, in people who had hypoglycemia unawareness. We've tested it, as I mentioned, in type 2 diabetes, in cystic fibrosis-related diabetes, and congenital hyperinsulinism. So you can see we've tested this device under various conditions, in various populations, different chronic illnesses, and we continue to see the same kinds of results. The system really is agnostic to what your condition is. If you're dysregulated, if your blood sugar is dysregulated, it's really most interested in just clamping your blood sugar and sort of following that. That is its goal. That is its mission. It's very single-minded in that respect. So with this system, we did all the studies you see on the screen. We've conducted these studies over the years. So we've done about since 2012, we've done 8 years, 7 or 8 years of clinical trials in the home use and outpatient setting, and we did this through a huge effort, campaign of raising funding into my lab at Boston University. Stephen Russell also had some grants that he got on studies that we were subcontract to, but these are just grants that came in through BU to fund all this work. The earliest funding came from the Coulter Foundation, where we started doing inpatient studies. But you can see this sort of campaign of writing grants, grant applications, and fundraising ran over 13-year period. We got money from the JDRF back in 2007 to 2010. The Helmsley Trust started supporting us in 2010, and the NIH in 2009. But all told, it was about $31 million worth of research funding that was used to do all this phase 2 clinical trials I've just shown you and reviewed with you. And so we just never could have gotten this done without these federal funding and private foundation and private giving. And then so about 20 million of the 31 million, so 2/3 of it came from the NIH. The Helmsley Trust has funded about $5 million worth of this work. But I really want to highlight, what you, the type 1 diabetes community, has done, and that's really this chunk here. So about 1 out of every $10 that we raised to do this research came directly from the type 1 diabetes community. That funding came into my lab at BU to help us build generally, most of that money went to building the Islet. The first, second, and third generation Islets were built. Most of the Gen 3, or some of the Gen 3 Islet was built in my lab, and some of it was taken over and finished by Beta Bionics. But a lot of that Islet development started, that Islet development, I should say, started in my lab, and these resources really helped make that possible. That would have been very difficult to get funding through traditional funding agencies. But the TMD community really stepped up, and we've raised a tremendous amount of money, and we're hugely appreciative of that commitment to this cause, and very, very humbled by it as well. So I thought it was nice to share that with you guys. But that brings us to Beta Bionics. And so we got to a point where around 2015, as I mentioned, we started building our own Islet, and it really that grew out of me looking around at the medtech industry and trying to understand how we can bring this forward. How can we build a system that's not a kludgy system, that's a cobbling together of an insulin pump with an iPhone and a CGM. Back then, at least, that just didn't seem a practical solution, to one where we could build our own small, compact, single wearable, purpose-built bi-hormonal system. Anything else really looked like it was going to be one, it was going to be 2 pumps that you'd have to carry, and it just was not really going to be practical. So that's when we started building the Islet, and it was in late 2015 that we decided to start our own company to build this purpose-built Islet, Bionic Pancreas. And we decided to incorporate this as a benefit corporation. My initial thoughts were either to do it as a benefit corporation or to do it as a nonprofit. We just didn't see a way that we could capitalize the company with the kind of money that would be needed to build a scalable solution and then bring that to huge numbers of people with type 1 as a nonprofit. So we ultimately looked back at this benefit corporation idea. And that was something that really was brought to my attention by my wife, Toby Milgram. She was looking at an article about benefit corporations back in 2011. They've only been around in this country for about 10 years. In 2010, the first states put in their state legislatures brought about these benefit corporation laws. And Maryland, I think, was one of the first states, and another state in the upper Midwest. But now we have 31 states in the US that have benefit corporation laws, Massachusetts being one of them. We particularly like the public benefit corporation law coming out of Massachusetts, and so we decided to incorporate Beta Bionics as a benefit corporation. And a benefit corporation basically is a for-profit entity You can raise money from investors just like a traditional C Corp, but it asks, it mandates, really, it requires that you have a benefit mission. So you have to articulate in the corporate bylaws and the articles of incorporation some kind of a benefit mission. Our benefit mission is all about protecting the interests of people with type 1 diabetes. And I'll write this down, and I'll put this up on screen in just a minute. But ultimately, the benefit corporations are structured such that they consider more than just the shareholder. There are other stakeholders that have to be considered when carrying out these companies' corporate responsibilities. In our case, we prioritize the benefit mission ahead of all other stakeholders. Our benefit mission, as you can see here, is to provide these turnkey solutions for autonomous glucose control to people with diabetes, bringing this technology to as many people with T1D as possible, continuing to innovate and offer the latest advances to the T1D community as these technologies improve. We want to bring that to people. And then the overarching goal of this company is to act in the best possible interest of the T1D community. And the T1D community is our first and foremost stakeholder. And as a benefit corporation, we're protected under Massachusetts Commonwealth law to be able to prioritize that stakeholder ahead of all other interests. And that's what we've done. This is our first and foremost priority. And so we have this commitment that is really codified in our public benefit mission. And as such, we're really committed to a 2-hormone system, a system that has this turnkey simplicity where you just type in the body weight and then let it learn and adapt evermore. And we started by raising some money from strategic investors before going to institutional investors. And with that additional initial capital, we took the Gen 3 iLet system that was being built in my lab, and we took that over and brought that into Beta Bionics and built this investigational system, which was the first purpose-built dual hormone system to be tested in human trials by us or anyone. And this device we began testing in 2018 in the insulin-only configuration. We did these 3 studies you see listed here. And in the interest of time, I'm just going to kind of go through these very, very quickly. The first study we did in the second half of 2018, and basically we recapitulated with this iLet system, this purpose-built system, everything we were seeing with our single hormone bionic pancreas that we were using in my lab at BU, except in a compact purpose-built device. The mean glucose levels we saw when the system was using either Feeis or Lisboa or Aspart was comparable to what we saw with the original Bionic Pancreas system in the insulin-only configuration. The mean glucose was about 155 mg per deciliter, with about 0.7% of the time below 54 mg per deciliter, so less than 1% of the time below 54 mg per deciliter. And the time in range was typically about 71% time in range, and we'd see about 40 to 50% of people on the device would have achieved the 88 goal for therapy if we'd run the study for longer. These experiments were typically on the order of 5 to 7 days in duration, and each subject spent a week on their own usual care, as well as 1 or 2 weeks on the iLet. So we didn't tell the device in this particular experiment about Feeis. We didn't tell it was a faster drug. We've since done a study where we told the iLet that the Gen 3 iLet study was a Novo Nordisk sponsored study where we told the device that it was using Feeis and configured the insulin absorption kinetics in the device to be aware of that, and we got better control when we did that relative to using just Lisboa or Aspart in the normally configured way. This was also the first study where we brought people with multiple daily injection therapy into the study. A third of this cohort, those shown in red versus the curves that the lines you see in blue on this plot, are people who are on MDI therapy who didn't necessarily have to have any experience ever using an insulin pump. And we did not need to wash out the insulin for these subjects, nor did we need to do anything more than type in their body weight to get them started. So that's really quite remarkable when you think about transitioning to a pump therapy. This system, we transitioned the MDIs onto the iLet just like the pumpers in the same way. We typed in their body weight and we sent them home. And there was no difference in the control that the device was able to achieve for those subjects who were on MDI therapy relative to the ones on pump therapy. And you can see here just an example of one subject on the device. This is the most poorly controlled subject on their own usual care. As you see here, the blue shaded region would be a mean glucose. The bottom of the blue shaded region would be mean glucose of 70 mg per deciliter, and the top would be about 154. And you can see there's a lot of variability from day to day on the average glucose that's shown above here in the white region. They're never hitting the blue line from day to day. This person not meeting goal for therapy on any single day. Their mean glucose was well above the desired goal. on days 1 through 7. But when you put them on the iLet, and you know, they're working hard to get this result, and they're still getting poor control despite their efforts. But when you put them on the iLet, of course, you see this person gets much better mean glucose control almost every single day. They're landing in the shaded region, and there's very little variability from day to day. And if you extend this same kind of plot to the whole cohort in that bridging study, you see there's a lot of variability from subject to subject, a lot of variability on any given day across the subject cohort. But when you put them all on the device, and they may have to do much less work, you know, on the iLet, of course, and you put them on the iLet, you get this tightening of these lines, much flatter curves, and many more of them are falling in the blue shaded region most of the time. So you're seeing about 50% of people in this case hitting the goal for therapy. That increases to about 90% when you look at the bi-hormonal system. And that's the study here. So in the middle of last year, we used the Gen 3 iLet. This was the last study we did with the Gen 3 iLet before retiring it. And what we see here is that these subjects were able to wear the iLet for a week in the insulin-only configuration and a week in the bi-hormonal configuration. In this trial, we didn't have a usual care arm. So they did these two arms in random order. And in this case, we used Humalog or Novolog for the insulin chamber, and we filled the glucagon chamber with this dasiglucagon. As I mentioned, it's 4 mg per ml. So it's 4 times more concentrated than the regular human glucagon. So the little 1 ml glass cartridge that we use to put glucagon in the iLet lasts about a week in the average adult based on our clinical trial data. And you can see the mean glucose dropped from about 150 mg per deciliter down to about 140 relative to the insulin-only system when you add the dasiglucagon. Again, that standard deviation around the population mean is quite small. As you can see, it's plus or minus 13 with the insulin-only, plus or minus 11 with the bi-hormonal, pretty much indistinguishable. The hypoglycemia, those cut about in half. That didn't quite achieve statistical significance, but again, it was a small cohort of only 10 subjects. So it's possible that if we had a slightly larger cohort, that would be a significant result. But nevertheless, it dropped nominally by about a factor of 2. So we again see a similar and in-line amount of hypoglycemia with the bi-hormonal system that we saw with the regular human glucagon studies we did with the old iPhone system. Mean glucose was totally in line with the old iPhone system, 150 for the insulin-only, and 140 for the bi-hormonal. About 71% time in range for insulin only versus 79% time in range for the bi-hormonal. And just as we've seen before, typically with the insulin only iPhone Bionic Pancreas, we saw about 50% of people would hit goal for therapy in that configuration, but in the bi-hormonal configuration, we were seeing typically 90% would hit goal. It's exactly what we saw in this study. So the dasiglucagon really stood up and showed us that it really does seem to be performing like regular human glucagon does, but it does so with you know, having this tremendous advantage of being really a scalable solution for managing you know, chronic you know, for managing hypoglycemia in this application, because it is as stable as, you know, Humalog and Novolog in a pump setting. So it's, you know, a couple of years in the supply line under refrigerated conditions, it's stable under those conditions, chemically stable, and you know, weeks at body temperature. So it's very practical to use in a pumping system like the iLet. So I just wanted to finish by just showing you where we are with the Gen 4 iLet and giving a little update on the clinical trials. I think what I'm going to do is, just in the interest of time, I'm going to zip right through to the end here and just show you what you know, what the clinical trial sites are that we're looking at. So this is, you know, the Gen 4 iLet. We've been putting that system together, and we're really close to bringing this to our first really phase 3 trial, which will be the first study we bring forward to the FDA for a market application for you know, for approval to then provide this device commercially to people in the insulin only setting. And we're going to do this through a phase 3 clinical trial that I've been talking to many of you about. Some of you may have heard you know, learned a little bit about our distribution of sites across the U.S. This is a map across of the lower 48 showing the 16 clinical sites that will test the Bionic Pancreas. This is the insulin-only Bionic Pancreas pivotal trial, but we're actually going to be using these same sites for the bi-hormonal Bionic Pancreas trial as well. And you can see the star down at the bottom is in Tampa is the Jabe Center for Health Research, which is our national coordinating center for all 16 sites. And so these sites are now trained and ready to go. And the training for this happened in in March, on the 10th and 11th of March, we were supposed to go down and have a have an insight training with about 70 investigators. So 3 or 4 investigators from each of the 16 sites were going to converge for 2 days in Tampa, where we were going to do all of our official training and get them ready to start recruiting. And unfortunately, COVID happened, and we ended up doing that remotely. So we had 75 people in the Zoom window get trained over a 2-day period. So we had 8 hours of training that happened on the 10th and 11th of March. But with that, all the sites could then become certified and begin recruiting. And I'll talk about that in just a minute. I just want to let you know that this is what the study looks like. There'll be 440 participants, 6 years and older with type 1. 110 of those will participate in an extension study. The study basically has a 3-month, it has 2 studies, a main study and an extension study. The main study will have will be all 440 participants. 4 out of 5 of the adults will randomize to the Islet, and 1 out of 5 would randomize to usual care for 3 months each. And then 2 out of 3 of the pediatric participants would randomize to the Islet, insulin-only Islet, and 1 out of 3 would randomize to usual care. So we're looking at the peds group 6 to 17 and adults 18 and older. And then for those that randomize to usual care, adults and peds, after the main study, they'll be able to participate in what we call an extension study, which is also is 3 months in duration, where those subjects, those 110 usual care subjects, will be able to participate in the study in the extension study and wear the Islet for 3 months. So everybody will ultimately get a chance to use the Islet for 3 months in this study. So it's a 13-week comparison study, but it's a 26-week duration because the usual care folks have to cross over into the extension study. A third of this cohort will be at least a third will be multiple daily injection users, and at least a third will be pumpers. And we're going to require that at least a third of the cohort have an A1C above 8, and at least a third have an A1C below 8. And so I just want to give you an update on where we are. Of course, when COVID happened, that really shook things up quite a bit, because we were planning to screen people in person through in clinic visits. And so we went back to the IRB the week before last and asked the IRB if we could do a verbal consenting through virtual visits. And we just got approval at the beginning of last week that the IRB gave the 16 clinical sites approval to begin this verbal consenting. So we're actually going to be able to begin virtual visits. And the great news is that verbal consenting in the 440-person cohort begins this week. So that was really great news. And we were very happy that we were able to make that happen. This study has been up on clinicaltrials.gov. The register's been up there for this particular trial for a few months now. The 16 sites in the JAPES have received many inquiries from interested potential participants. So they've really put together at each of these 16 sites a pretty good list of participants. So I would ask that you not overwhelm the clinical sites at this point with petitions to participate, because over the past several months this has been going on, and these sites have identified many potential participants. So right now they've got to focus on the recruitment, and our goal is to get all of the verbal consenting done over the next couple of months. So that's where we are with the trial. Of course, there is some thought that there's some concern that the trials are that they're thinking, the clinical sites are thinking that realistically, we're going to ultimately have to bring people in to put people on the Islet, and for that to happen, people will have to come into clinic. That's at least the plan right now. So that certainly won't happen until it's safe to do so. So ultimately, the goal would be to do that probably, thinking the earliest that could happen would be around July at this point, is what our clinical team has been suggesting. So we'll just have to see how the crisis evolves over the course of the next several weeks to understand better about when we can actually bring people into clinic to get them on the device. Right now, at least, we can begin the consenting process. So with that, I think I just want to finish by pointing out that this has been an effort of many people's work over 18 years, really, when you think about when this all started, and the animal studies and human studies and developing the device and so forth. This is a picture at the Friends for Life conference a couple of years ago, 3 years ago, I think it is, of our team. We had about 18 people for a while there, and then ultimately, we've grown the group. We have about a little over 40 employees now at Bionic, so we're really a very small, still quite a small, but rather energetic group. And you can see a picture from Friends for Life last year of the team, as it looked most of the people were represented there at that point. We're a little bit bigger now, but still a pretty small group. And then finally, the broader group beyond just the folks at Beta Bionics that made this possible really extends across, folks who worked in my lab over the years at Boston University, with Stephen Russell, at MGH, at Stanford, and University of Massachusetts Medical Center, and University of North Carolina, and the Moores Clinic, and University of Colorado, and so forth, in the summer camp, Claire Barton summer camp studies that were done there. And all these industry collaborators, which were so important to moving various parts of this technology forward over the years, from the laptop system to the iPhone system, and then ultimately building out the iLet system with our collaborations with Dexcom and Senseonics as well. And finally, of course, the research support that I already alluded to was so important to making this happen, with tremendous commitment from the National Institutes of Health, but also from the Helmsley Trust and the private giving that came to my lab through the type 1 diabetes community directly. So I thank you guys all for your attention, and we'd be happy to take any questions.
So thank you all. We had quite a crowd, and most people stuck to the end. Ed, we have an enormous number of questions, and what I would like to suggest is I'll pose a couple here that there were multiple people asking, but we have all of the questions, and Ed can answer them afterwards, and we'll put a PDF online associated with this video with the answers to all of the questions. So if we don't get to yours right now, you'll get an answer. Give us a couple of days. So one that we heard a couple of times in here is, what happens if there's a CGM failure on your device?
That's a really good question. So the way the system works is, we have multiple insulin controllers that are running continuously and in parallel, sort of simultaneously all the time. We have one that's really responsible for meeting your basal metabolic insulin need. We have one that's responsible for correcting hyperglycemia as your blood sugars leave range. And then we have a third controller that learns about what typical meals are, how much insulin it needs to deliver for typical meals at breakfast, lunch, and dinner. And these three controllers work together in a very harmonious way, every 5 minutes reevaluating and assessing how much insulin to give. But as they do that, they're also keeping track of the amount of insulin they give in these three different parsed out ways, so that if ever the CGM is offline, the device has essentially stored and is constantly updating every 5 minutes its best guess at your basal rate, your basal insulin rate. So if the CGM's offline, we basically have 288 different basal rates every 5 minutes. There are 288 5-minute steps in a day. So the device is accumulating what your average basal delivery is at 12:05 p.m., at 12:10 p.m., at 12:15 p.m., and so on. So it can invoke that basal rate immediately if the CGM goes offline. If your blood sugars have excursions above normal range, you can enter a finger stick measurement at any time when the CGM is offline. And the other controller that's responsible for handling hyperglycemic excursions can then give a dose automatically in response to that big BG. And then finally, if you have a meal when the CGM is offline, then you can just use the meal announcement feature that is very simple to use. You're not counting carbohydrates, you're just estimating if the meal falls in a usual, more than usual or less than usual size in terms of carbohydrate content, and you use it exactly the same way, whether it's used as with the CGM online or CGM offline. And then a couple of hours later, you'll be warned to check. You'll be encouraged to check a blood sugar, in which case it can add more insulin and correct with the correction controller. So all three controllers are kind of running all the time. You never go out of closed loop in our system. You're never thrown out of closed loop. There's no other mode but closed loop. It's just that when the CGM is offline, you have more sparse data, which is whenever you enter a BG, but the system can then act and respond to that whenever a BG is provided.
Very good. Another question is, if I'm taking other diabetes medications such as Victoza or metformin, how will this system work?
That's a very good question. I mean, we haven't studied it in the context of those other drugs. However, we do intend to look at that in the future. We want to understand how well the system works if you were to add drugs that could increase insulin sensitivity. How well would the system work if you were to add a basal insulin? If you have somebody with type 2 diabetes, for example, who might want to use their basal insulin, and then the iLet would then give presumably about 50% more insulin or so for them, for their prandial insulin and so on. So these are studies that have to be done. But I would say that because the device is so inherently adaptable, because the amount of insulin dosing it can give can adjust really quite dramatically from person to person, and even within the same person, to respect the intra-subject variability that is a part of diabetes management today. I'm optimistic that if you bring other drugs on that could influence insulin sensitivity, the device could account for that and accommodate it. But again, that's going to be the work of future. That's going to be this, study of future work, and that's something we're certainly going to have to and want to do.
All right, I'm going to ask 2 more questions, and I'm going to get to the most asked one is, when is this going to be ready?
That is the most asked question, of course, and there is some uncertainty, of course, in the light of what's going on around us today, right? I mean, with COVID outbreak, it's really it does put a hamper on things. But if we can, I can sort of say best estimates would be that if we can actually get the people into clinic in the summertime, in July or August, and get people on the device, then I'm hoping that the timeline doesn't slip by more than a couple of months. 1 to 2 months is what we're hoping from all of this. If that's the case, then I think it wouldn't be unrealistic to suggest that we could have the device ready for the FDA to begin reviewing it by the end of the year still. We were hoping to get the device in front of the FDA by the end of October, beginning of November, and if we can protect a submission that's 8 weeks later than that by the end of December, we'd be pretty happy. Very happy with that, given all that's been going on. I would say that the FDA has been really, really fast and responsive to reviewing these applications. They've typically reviewed applications for hybrid closed loop systems at least in about 90 days. This is a de novo device. So it's the device, this purpose-built device has never been looked at by the FDA, this particular version of the iLet. So they've got a lot on the plate, but I think they're still committed to doing it in 90 days. And so if we can get it to them by the end of the year, then I'm thinking by the second quarter of next year, we could hopefully have approval. We did receive breakthrough device designation by the FDA for this device, and we hope that will help continue to prioritize it among the applications they're going to be receiving in the next year or so.
Okay, and then the last question, we've got several attendees from outside the US, and they were asking about, when do you anticipate, or do you have plans for availability outside of the US?
That's a really good question. So it's certainly a high priority of ours. We want to get this out to as many people as possible, and that means everyone with type 1 diabetes, not just people in the US. We have been talking to people in the Scottish government and folks in Australia who are connected to people in the Australian government to try and start those conversations about bringing this device into other markets. So we're really committed to that. What that will require in Europe, at least, is a CE mark as opposed to a 510K clearance that we would get here in the US. The CE mark is a process that takes 6 months or so. I'm not sure how that is all going to be impacted by COVID, but our plan was to submit the CE mark application this year, and I think we're still planning to do that. If that's a 5 to 6 month process, we don't know, but in light of what's happening, but hoping that can happen in this calendar year, so that around the same time we could launch in the US, perhaps we could have a CE mark. Of course, having a CE mark is not the same thing as being available in all these countries. There's still procurement in each of these countries, in Europe and other markets, and we'll have to navigate all those things. So we're a small company, as you know, and we're going to do everything we can, but we're very committed to try and move things into other markets outside the US, next year.
Very good. Well, I will make sure that Ed gets all of your questions, and he can answer them while he's sitting at home enjoying whatever he enjoys while we're all cooped up. Thank you all again for joining us for our first virtual Friends for Life conference. At the end of this, you will get a 3-question pop-up survey. Please take a couple of minutes to give us your feedback. I'd like to thank again all of our staff, all of our speakers like Ed, who spent today helping us deliver this. And a big thanks again to our sponsors, Lilly Diabetes, Dexcom, Insulet, Tandem, and the American Diabetes Association. A video of this will be online within a day or so. Everyone, be well. Bye-bye.
Thanks, Jeff.